And how many chromatids is this over here? 2. So if this was from the mom and this is the mom's copy post the synthesis phase, this would be the dad and the dad's copy, correct? Now, are these 2 sets of chromatids, or are these 2 chromosomes, are they identical? But they should be coding for the same stuff. Hair color, foot length, whatever it is. I don't know. But meiosis 1 separates out these homologous chromosomes. So if this one was from the dad and these 2 were from the mom, in other words, if your body was making gamete cells, which you being made of both parents, if you were making these gamete cells, then the first thing you would do in meiosis 1 is you would split right down the middle. So you're separating out the homologous pairs. The second cell division, though, which is called meiosis 2, would result in the sister chromatids separate. Now, we've seen the sister chromatid separation happen before. Where did we see that at? Mitosis. So I always like to tell people that sister chromatid separation event, if you want to know more about that, reference mitosis. Now going back up to my picture, if I was to observe a splitting of the chromatids, that separation of sister chromatids would have to facilitate a division right here and a division right here, which is just like mitosis for all practical purposes. And the end result has to be how many haploid cells? 4. So we know that after these 2 divisions, the end product is 4 haploid cells. Each having half of the DNA. And for humans, how many chromosomes would be that half of the DNA? Yeah, 23 chromosomes. So showing you a picture and observing this picture on the board, I guess not. Okay, there we go. How many chromosomes are in this topmost cell, the parent cell? 2, right? 1 and 2. We consider the blue from the dad, the red from the mom, whatever you want to. And how many chromosomes are in this post-replication? 2. Now, a lot of people would say, well, when you replicated, if this was 1 and 2, then why is this not 1, 2, 3, and 4? Well, because those 2 are identical, and those 2 are identical, right? Okay. And then, what are you observing in meiosis 1? If these are called homologous pairs, it seems to me that meiosis 1 has resulted in their splitting, or their division. Now, what's happening in meiosis 2? What do you appear to be splitting in meiosis 2? Now, let's talk about just some logic real quick. How many chromosomes are in this cell? How many are in this cell? How many are in this cell? 1. How many are in this cell? How many are in this cell? So let's put the words haploid and diploid on it. Haploid or diploid? Haploid or diploid? This one. Haploid to the power of 4. Okay, sweet. So you become haploid after meiosis 1. Now, from that, sure, when do you observe the replication of DNA? When does that happen? Before meiosis 1, after meiosis 1, after meiosis 2? When? before. So we might say that meiosis 1 is preceded by a round of interphase, which we know interphase as being a G1, an S, and a G2 step, and that G1 and G2 are the growth phases, whereas the S is the synthesis phase, which of course is what results in the production of the sister chromatids. Okay, simple enough. Now let's talk about some of the major events that's happening in all of this. I'm going to talk today about the events of meiosis 1 in the greatest detail, because meiosis 1 is the newest if you think about it. Now, why do I get to avoid going through a large description of meiosis 2? Do you know why I get to do that? Because it's mitosis. So when someone says, well, but what about meiosis 2? My answer to them will simply be, just think about mitosis and you got it made. So let's talk about meiosis 1. And to start us off, let's talk about the most important event of meiosis 1 and the most important event of all of meiosis, which is the first prophase, or prophase 1, which of course is in meiosis 1, hence the one after it. Between both versions of meiosis 1 and 2, 90% of the time is what it takes to get through prophase 1. So prophase 1 is 90% of both meiosis and meiosis 1 and 2. It's a phase in which we observe the condensing of the chromosomes, which we've noted before in prophase. But it's also a very important time in which you observe a synapsis. This is where your chromosomes, the two sets of sister chromatids, the homologous pair, line up. And it's here that you can observe crossing over. Now, when you observe crossing over, what happens is the non-sister chromatids exchange information. This makes each chromatid uniquely different from its sister chromatid. The four yes. That's like homologous chromosomes. So one from the dad and one from the mom exchange information. That's correct. Yes. Gene for gene, they line up. These four chromatids that are all together constitute something called a tetrad. And the crossing over event is something that happens at the chiasmata. So if you observe these two pairs of sister chromatids, and then you notice these two pairs of sister chromatids, how many total sister chromatids do I have that have lined up? 4. And these two pairs of chromosomes are called what type of chromosomes? Homologous chromosomes. And since you have 4 chromatids, so I'm going to call that a tetrad, and if I exchange information between two non-sister chromatids, then that would be creating a synapsis at a chiasmata. So here's what we're going to observe. We're going to observe that this section and this section are going to cross over. So after the crossing over event has happened, we're going to be left with something like this. Keep in mind that whenever you had created the sister chromatid, after you'd done the synthesis phase before meiosis 1, you had two sister chromatids that were identical. Those two are identical, and these two are identical. Now, tell me what crossing over what has it done? Are these two sister chromatids that I'm pointing to, are they now 100% identical? Are these identical? So that means that when you're making gamete cells, when you're doing meiosis in your body to make either an egg or a sperm, or any creature for that matter that's in the animal kingdom, any creature that does this is going to do crossing over, which, if you think about it, takes the parental chromosomes, the chromosomes that were given by your parents, and mixes and matches them. So now that you are not just giving a unique unit of one parent, you're giving a unique new unit of both of your parents to your egg or to the sperm. Good? Okay. Now, a question that I have for you is, why go through this trouble of making your chromatids now a little bit different from each other? Do what? I think I heard someone say it. It's going to increase the variation. You have new mixes and matches, new little combinations of traits that didn't exist before. So this increases variation. And again, that's called crossing over, which happens in prophase 1 of meiosis 1. Now, after prophase 1 is the standard rest of the PMAT, the metaphase 1, the anaphase 1, and the telophase 1. Metaphase 1 is when you line up the homologous chromosomes at the equator. Or the metaphase plate. And what are you splitting up in anaphase 1? Are you splitting up homologous pairs or is the sister chromatids? The homologous pairs. So in anaphase 1, you observe the splitting of the homologous pairs. And in telophase 1, you reform the two nuclei around each set of chromatids. And of course, then you will begin, shortly after that, the cytokinesis of part 1, followed by prophase for part 2. You do not re-enter a G1 or S or G2 phase. So I'll write after this that you continue into cytokinesis 1, and then prophase, metaphase, anaphase, telophase for part 2, and your result would be 4 haploid daughter cells. Yes, Alex. In each cell, after the telophase 1, after this event here, after you get to this point, you should have 23 sets of sister chromatids, or pairs of sister chromatids in each haploid cell. So meiosis 2 will split those sister chromatids up so that there is 23 individual little chromatid / chromosome units in each of the haploid cells. Okay. I have another picture I want to show you real quick. And I want to just discuss the implications of this picture, and then we'll be close to done. Okay, so this picture right here shows you a section of anaphase and telophase from meiosis part 1, right? What does it appear to be splitting in that anaphase 1? Are you splitting the sister chromatids here, or are you splitting the homologous pairs? Okay, very good. The splitting of the chromatids happens in meiosis 2. That's for tomorrow, obviously. Now, let's assume that the dad is... Oops. Let's assume that the dad is the red color. No. No. Yeah, okay. Let's assume that dad is the red color and the mom is the blue color, or whatever you want to. Do you see all the red ones, all the red chromatids going to the north pole of the cell? Do you see all the blue ones going to the south pole? So, and have I led you to believe that these two could possibly not reverse their position? I think that they possibly could. So what does this mean? This means that these chromosomes, when they do divide and when they split, they do so independently. In other words, not all the red chromosomes or the paternal chromosomes have to go to the north spot, and not all the maternal ones have to go to the south. They're intermingled. So that means that these chromosomes segregate independent of other segregations. That is going to be the second time today we have observed an increase of variation. So to keep in mind about increasing variation, obviously its importance can't be understood. In other words, I think everyone knows what increasing variation is going to do. It's going to create unique little packages of creatures that might be selected for in an ever-changing environment. But increasing variation has happened through two ways as of today. The first way we observed was the event called crossing over, which was in prophase 1 of meiosis 1. The second event is going to be the event called independent assortment, which is what you just observed, essentially. The fact that these chromosomes independently segregate regardless of type. There's going to be a level 3 reason for increasing variation, but we'll get to that tomorrow. Any questions about what you see? Okay, to help us understand this, I have a fun-filled but yet action-packed video that I found on the internet when I was y'all's age and I took AP Biology.